actually going to be the last official seminar of the year, and then there will be the second annual ice cream field trip 2 weeks from now. But we're very pleased today to have Dr. John Gore with us, all the way from Nashville, Tennessee. Dr. Gore is a very well-known figure in the MRI community. He holds many different appointments at Vanderbilt. He's the director of the Institute for Imaging Sciences. He's a professor of radiology and radiological sciences, biomedical engineering, and I believe holds appointments in a few other departments. And also is the Hertha Ramsey Kress Chair of Medicine. His other accolades are too numerous to list out, but some of the highlights are that he is a gold medal winner of the International Society for Magnetic Resonance in Medicine, which is basically that society's highest honor. And he is also the editor-in-chief of the journal Magnetic Resonance Imaging. So very pleased to have him here today to talk to us about some of the things that are going on in this lab. Good. Yeah.
Well, it's a pleasure to be here. I'd like to thank Melanie for the invitation. There was no mention of the ice cream trip. I mean, that sounds...
You could come back.
I could have come back a couple of days. How many people here actually have no idea at all about how MRI works? Oh, good, because I was told there might be some people who aren't that familiar with MRI. I'll try to explain stuff as I go along. But I'm going to talk about some of the work that's kind of a general topic, but one that we've been heavily engaged with for a number of years, looking at different ways to get contrast and information from images. And if you think about MRI, we've had over 30 years now of steady progress. It's arguably the single most important, useful technique in medicine, especially if you're looking at like soft tissue pathology, strokes, tumors, that kind of stuff. It produces these wonderful three-dimensional images. On the left there is a fly-through of a brain at 0.6 millimeter isotropic resolution. That was recorded at 7 Tesla a couple of years ago. Very nice detail. And you can also make images of multiple different properties, non-invasively. And as we, engineers and physicists continue to make big improvements in image quality, in reducing cost, in ease of use, speed, etc. Just to verify that I'm actually hardly more than 30 years old. This was an early MRI machine. It's actually the first MRI image I ever saw in 1976. It was of a dead mouse in Aberdeen, in Scotland. It was taken at a field of 0.04 Tesla. You can tell it's dead. They broke its neck, and this is the edema that's formed at the region of the brain. So this was the very starting point of MR imaging. When I went to Yale in 1984, we were very happy to get images like this with several millimeter resolution in about 8 minutes, and that's an image of the brainstem. And then this is what we did. It's already 7 years ago. This is an image of the midbrain, same structure, just much better resolution, much better signal-to-noise ratio, much better contrast. And that's been the progression. That's why it's been so successful in medicine, because we can really get exquisite images with breakthroughs in technology. In addition, the kinds of information we get from MR has continued to increase as well. In fact, one of the things that I always tell students originally is that there's no such thing as an MR image. You say you do MR imaging, but in fact there's dozens of subtypes of MR image, and they're distinguished by basically their contrast mechanism. And the contrast mechanism is really any process, whether it's a physical process or a biological phenomenon, which modulates the signals that you use to make the image. And so those can be of different types. Here's just an example. These are all MR images. Top left, those depend on things that are called relaxation times, T1, T2. And they are useful because at the top there you can see that we have, I'll use this. We have a tumor that shows up as different in relaxation. We have an MS plaque. These is an MS plaque. So these differences of gray and white have to do with relaxation time. This is a map of blood flow. This is the Gore signal used for functional imaging. This is diffusion imaging, which is telling us about a stroke in this area. And it is anisotropic, which is white microstructures. These are veins, these are metabolites, this is mild water. Just a whole range of different kinds of information that you can So what I'm going to do today is just revise one elemental aspect of the physics of NMR so that everybody starts out with a level playing field. Then I'm going to talk about two kinds of contrast. One which is basically produced by applying magnetic field gradients from the outside, and another one which is to do with intrinsic gradients. So that's the theme that connects the two, that whenever you have magnetic field gradients, that is non-uniformity in the magnetic field, you can get information about microstructure using diffusion, and you get functional information and other things using gradients. And the real question that drives this is, can we get new kinds of information about tissue structure, particularly below the level of the voxel? So a voxel is maybe 100 microns or a millimeter in size, but you know this stuff at a lower level. There are cells, there are structures much below that level. Can we learn about those even though we can only sample at a larger scale? Or can we get information on, say, physiology or whatever? So that's where we're going. And just NMR physics in brief. We're talking about water molecules in tissue. We're talking about protons into hydrogen in water molecules. Their nuclei would spin a half. They correspond, they have a magnetic dipole. In the absence of any applied magnetic field, they point randomly in all directions. We apply this very strong external magnetic field. It causes the energy differences between pointing in different directions to be different, and most of them prefer to point up rather than point down, and that means we have most of them pointing in one way, so that gives us a macroscopic magnetization, which is the thing we measure in all NMR experiments. So just from putting your water in your brain or wherever into a magnetic field, you will induce this magnetization. The remarkable thing is, because it comes from nuclei with spin, if you is pointing here, if you inject a rotating magnetic field, so-called B1 field or radio frequency pulse, that goes around at a particular frequency that's proportional to the field, then you can cause this magnetization to gradually tip away, and it will gradually tip down into the transverse plane. Then you can turn the field off, and that magnet is now, because of its angular momentum, its spin will rotate around. So if you wrap a coil around the sample, you'll get a signal. So the whole idea is you stick a sample into a magnetic field, you inject a radio frequency pulse at some frequency that's selected, you turn it off, you get a signal in a coil that is just induction from a magnetization within a conductor by Faraday's law. So there's two equations in this talk. I was told I could have at least one equation, but it's engineering, not radiology. Radiology, if you put the election in, they just throw you out. It's just instant death to the seminar, you just have to leave. Here it is, whoa. So this is the signal that you induce. It's oscillating at a frequency that depends on the resonance frequency, that depends on the field, depends on how many spins you have. It also decays with a time constant, normally written as T2 or T2 star, because gradually over time that whole phenomena kind of gives up and you lose what's called phase coherence. The spins relax back to equilibrium until your signal goes away. And this is one of the time constants that people measure sometimes, and they use that to vary the contrast in an NMR image. So, that's the basics. I thought I'd start out talking about this process of diffusion, mainly because this was the topic of Professor Martin's PhD thesis when she was at Yale. And we've recently gone back to re-examine some of these ideas. But the idea is that the NMR signal, that decay rate, is actually sensitive to the motion of the molecule, to the water molecules. And in particular, if they diffuse around, random motion in different directions, if they do it in the presence of a magnetic field gradient, that is something that varies the magnetic field from place to place, then those incoherent motions causes a signal loss. It's a kind of transverse decay, just like T2, because the spins gradually get out of step and out of synchrony. And this was first observed many years ago, 50, 60 years ago, by Hahn and Torrey. A particular method was devised by these guys, Stitch and Tanner, and it's been used to measure the diffusion of molecules in liquids for many years as a standard kind of measuring technique. In imaging, we make use of it to modulate the signal. I'll explain how shortly, but here's some examples of how it's actually used clinically or practically. So for example, in a stroke, this is a region of ischemia ischemia. Minutes after the stroke occurred, immediately you see the signal is brighter because the diffusion rate there has dropped. So spins are moving less freely there than in the surrounding tissue. Because of that, we get a signal difference. If you apply the gradients that we're going to talk about that go into different directions, you find the diffusion is not the same in all directions. In particular, in white matter, which has a kind of tubular structure, a tract structure, the diffusion rate, the ability to move along the white matter tracts, is much easier than to go across where there are membranes in the way. And so therefore we have anisotropic diffusion. But by using gradients in different directions, we can actually construct these nice images like this, which show the white matter tracts within the brain. This is being used a great deal in both research and clinical activities. If you have diffusion where the cell density is not very high, it turns out the water within cells is not free to go in and out, so it's kind of in a little compartment, whereas the water outside the cells can move much more freely. And so in loosely packed cells, you have a fairly high value of the diffusion coefficient. This is a tumor, but as the tumor cells grow and proliferate and they fill in, become more dense, the diffusion drops. So this is actually being used clinically to say something about cell density in tissues and tumors in particular. So how does this actually work? Well, we know that water molecules wander about randomly in tissue by this process called Brownian motion. And you go to all these meetings where the psychologists now, they're all over diffusion and DTI, and they all talk about Brownian motion. And I asked them, who was Brown? Anyone here know? See? It's important to study the history. Brown was a Scottish botanist about 200 years ago. He was studying the motion of pollen in the air. And he actually talked about it being Brownian motion and began to devise methods to measure it. Anyway, in a free solution, after a time t, molecules start to move away from where they started, and they move on average, the mean square distance traveled from where they started is given by this expression. This was actually derived by Einstein. It's one of his 3 great papers of 1905. It's in proportion to the square root of the time, so that's characteristic of diffusion, and this constant known as the self-diffusion coefficient. So if you start a water molecule off at some place, you can predict where it's going to be at a time later according to that equation. But if you actually measure that in tissue, things don't get as far as they should because of hindrances to the free movement. So if you, for example, were to measure how far away a water molecule gets after you started off as a function of the square root of time, it should go up linearly because of that equation, and the slope would depend on the diffusion coefficient. But in fact, after a while, because you begin to bump up against restrictions of some sort, those restrictions can be other macromolecules, they can be organelles, they can be cell membranes, and that seems to be the big one, the cell membrane. Then you find that things begin to level off here, and in fact, after a while, they won't get any further because there's no, if they have if they're inside a little sphere or a volume with an impenetrable barrier, they can never get further away than the size of that barrier. So if you take the slope of this line, we get what's called the apparent diffusion coefficient. So the apparent diffusion coefficient down here is quite high, and it gradually goes down to 0 as we go to longer and longer diffusion times. So water in tissues is less than a pure solution because of these restrictions on free diffusion. That's the reason why we can do DTI, because it's more free going across the tract than down the tract. In practice, in most tissues, this ABC depends on the number of cell membranes that the molecules encounter and their permeabilities. And in fact, if you look at tumors, this is being used clinically. This ABC is actually interpreted as an inverse measure of cellularity, and it changes after treatment. So this is what we're talking about. I showed that earlier slide where if you have loosely packed cells and it's restricted by the size of the membrane here, but the stuff on the outside can travel more freely, this has a fairly high ADC. As the cell density goes more dense, the ADC will drop. If you treat this and it begins to go back towards this, the ADC will go back up. This was the slide I showed. So it showed that as the cells fill in, the ADC actually does drop. this is an example of how it's used clinically. This is a patient undergoing treatment for cancer before having surgery, and this is before the treatment, after the treatment. This is the map of the ADC. This patient responded, which means that they gave him a treatment. This was the ADC map before the treatment. After one cycle of treatment, there was an increase in the ADC of 12%, meaning you killed a bunch of cells and the water was now more free to move around. In this patient who was not a responder, the ADC continued to drop by 12% because the cells continued to proliferate and get more and more dense. So within a couple of days of starting treatment, here's an imaging technique that can tell you whether this patient is likely to respond, should you change the treatment, etc. This is kind of well established. The method that we use relies on this sequence. This is the original sequence from a Stechel pattern, the pulse gradient spin echo technique, where you apply a gradient that labels the phase of the nuclear spins. Then they start moving around. You wait some time, delta, big delta, you put on another gradient that should remove that phase evolution, but anything that has moved will be moving from one place to another, from one resonant frequency to another. It'll change its NMR phase. You'll get a mixture of phases. That means the signal decays. It decays according to how long you wait and how strong the gradient is. So this is the equation that says the signal will go down according to how long you wait in that situation. Now inherently, there's this intrinsic timescale in the measurement. That is, inherently we incorporate the measurement delta over which we're measuring that diffusion. It's actually very difficult to achieve extremely strong gradients for very short durations and not have much in between. We saw that the amount the signal goes down, and you have to have a signal reduction of a few percent at least. That depends on the gradient strength and the weight. And there's limits on the gradient strength, so the weight in practice has to be a certain amount. And in practice, all diffusion in the literature is measurements are made with this diffusion time being tens of milliseconds. Well, if you wait tens of milliseconds, let's say you wait 50 milliseconds, then mean-free distance traveled is about 20 microns. So in 50 milliseconds, the water molecule will get on average 20 microns away. Well, if the cell size itself is only 10 microns, then it only can get 10 microns if it's impermeable. So you can't learn anything about things on the size of a 10 micron or smaller scale. In other words, you're going to be limited by cell density, yes, but you can't learn anything about subcellular changes. If you want to probe things on a subcellular level, you've got to use shorter diffusion times, much less than a few milliseconds. And that's very hard to achieve by conventional methods. So the questions we began asking years ago when Melanie was a student were, what are the relative contributions of restrictions at different scales, and can we actually do something to detect changes at the subcellular level? So for example, when you treat a patient with chemotherapy, long before the cells die, they may undergo apoptosis, they may undergo some other changes. You would think there's going to be changes within the cell before the cells die and you see a change in cell density. So potentially, making measurements looking for intracellular changes could be an earlier marker of some kind of response. So this is the method that's being developed, which is we replace these two gradient pulses, which are just on-off type pulses, with oscillating gradients oscillating at a particular frequency. And the very nice thing about this is that you no longer depend, the relevant diffusion time is no longer the time between these pulses, but it's actually the period of this oscillation. So that we're not limited by this being 40 milliseconds. If we oscillate these gradients at, say, 1 kilohertz, then it becomes of the order of a millisecond. In fact, the effective diffusion time is given by 1/4 times the frequency. So if you go at 250 hertz, then your effective diffusion time is now only 1 millisecond. And that's something you can't achieve using conventional methods. So this is known as OGSC, or oscillating gradient spin echo methods. You replace these bipolar gradients with oscillating cosinusoidal harmonic gradients at a particular frequency, f. The effective diffusion time depends on 1/f. And that means if you go and say at kilohertz, your average distance over which you're sensitive to diffusion is much less than, say, a micron. It's much smaller than a cell. What you can then do is you can make the measurement at one frequency and you can sample structure at that particular diffusion time. You can then change the frequency, you can sweep through the frequencies and actually get what's called a diffusion spectrum, a temporal diffusion spectrum, and show that this measured diffusion coefficient varies with frequency in a way that tells you about the microstructure of the tissue. Now there's a whole background theory here, I don't really want to delve into it too much. For those who are interested, there are papers on this, but basically this diffusion coefficient that we've talked about as being a simple number, the ADC, it's really a function of frequency. It's a function of the frequency in our measurement. And those frequencies actually we can show are related to the distribution of correlated velocities in the molecular dynamics of the water molecules. And that depends on the structure because the water interacts with the structure. What the gradient waveform you select does, it tells you where you're going to sample this spectrum of diffusion. So I'll flash this. This is the second equation. You need to pay attention. But what they said is that the signal now depends not only on this diffusion coefficient, that's a function of frequency, but where you measure that frequency according to this function, which is dependent on the gradients that you choose. If you choose the gradients to be a single code sinusoid, then this becomes a delta function at some frequency. You just measure this at that particular frequency. But you can then vary that and map out the entire spectrum. This is actually the theoretical spectrum. This is what you could measure. But this is the theoretical spectrum. If you have impermeable spheres, closely packed, or you could just have one. You have a sphere, and you measure this apparent diffusion coefficient as a function of the frequency. And this is for different diameters. And what you see is that if you have very high frequencies such that the diffusion time is very short, everything is pretty free, there's no effect of the boundaries, so things are up here, and you're measuring the intrinsic diffusion of the solution. As you go down in frequency, you begin to drop the ADC because now you're encountering the restricting effects of the boundary. And the pulse gradient spin echo method is equivalent to measuring down here at very, very low frequencies. Where in fact there's almost no difference in these measurements for these different sizes. That's what we were saying, there's no sensitivity to the intracellular type dimension. But if we go to higher frequencies, say like 2 kilohertz, there's a big difference depending on how big the sphere is. So we thought finally we could talk about this for ages and never really proved it. So we thought we'd do some real proofs of concept. So we took some little microcapillaries of different internal diameters and we filled them with water. So this is just one compartment, close-packed cylinders filled with water, no water on the outside, and we measured the ADC against frequency over this range, 0 to 600 hertz, very easily achievable on an MR system. And these are capillaries of different diameter, and these are the measured points, and this is the fitted lines from the theory. And you can see it's extremely well behaved, and in fact if we take these fitted lines and we invert the expression, we can estimate the size of the compartment you can see here compared to the real one. Within, I don't know, a few percent, we're actually spot on in estimating the size of those compartments. So this is a way to measure aspects of microstructure in this very simple system we've proved is pretty exact. If I go back to this curve though, and I look at these dimensions, these are the dimensions of a lot of interesting structures in biology. And if I look at this region down here, and the region down here is much easier to achieve on things like human scanners. This whole region is achievable on a small animal scanner, but there's some other issues that go into humans because of gradient power, etc. But if I just blow that area up, I see the following, which is I have things that look pretty much like straight lines, right? And the difference between these straight lines is the gradient, and the gradient varies with the size of the cylinder. So we call this the dispersion, the diffusion dispersion rate, and we propose that therefore that's a simple measure, just to make two points, join a straight line between them in this regime of say somewhere between 100 and 150 hertz, and just point 2 points, measure that slope, and now you have a measure of the diameter of the cylinder. Well, it's not quite that simple because these curves do curve over, but if we went for that that if we said we measured, say, at 50 hertz and 100 hertz, these are relatively low, easily achievable frequencies. We just make that measurement of 2 points, and we plot the slope against the size of the cylinder. Then you can see there's a monotonic relationship until we get to about 8 microns, after which it begins to curve over. But over this regime, the 2-point measurement should be a pretty good estimate of the diameter of the restricting structure. And this would actually be a pretty good model for white matter, because white matter is a series of cylinders, essentially all aligned, particularly in places like the spine. So here we have the spine is actually a rack, and these are different regions of interest within the rack spinal cord. And you can see they're labeled here. I forget what these names stand for, but they stand for something technical. And then these are different histological sections. You can see there's a variation in the axonal diameter, according to which of these areas we take. And when you look at the measurement of the diffusion coefficient as a function of frequency, you can see they actually have nice straight lines, pretty good R values, but they have different slopes. If you look at how these are behaving, if you go to diffusion time of, say, 140 or 58 milliseconds, you can see there's no contrast at all between different parts of the white matter. But if you go down to these short times, like 1 or 2 milliseconds or thereabout, you begin to see contrast, which is telling us that, in fact, this technique with very short diffusion times is truly sensitive to the differences in the sizes of the restricting compartments. And so doing those fitting curves, again, we can end up with this. So we're saying, using a 2-point fit, and we get this in just a matter of a few minutes, we can actually get a map of axonal diameter in the white matter of the rat spine. And in fact, we've shown this correlates very nicely with histology. And then in the human, you can segment out the corpus callosum. We claim, and we've not validated this, but in 3 minutes at 3 Tesla, we can get a map like this or this, and actually quantify the distribution of axon sizes throughout the structure in which those cylinders are all aligned. So this may be one way to actually approach rapid measurements of in vivo axon diameters. You can extrapolate this to other cell sizes, which is a little bit more tricky theoretically, but it's a promising method that's got some practical translation. If I go back to this slide, again, look at how these different size structures behave. Let's say I measure at 0 or low frequency, they're all about the same in terms of the ADC. But if I go out to say 100 even, or let's say 1,000, you see there's a big difference in the ADC. So what that would mean is if I make an image in which I oscillate the diffusion gradients at this frequency, I'll get a very different signal strength from structures of different size. So where does that play a role? Well, we showed a couple of years ago that these are images of a rat brain with a tumor. This is the conventional proton density weighted image, depending on T2. This is the conventional pulse gradient spin echo measurement. And you can see there's a change on the tumor side compared to normal. And then these are different measurements taken with increasing frequencies. And by the time you get up to, say, a couple hundred hertz, you would actually begin to see, instead of a heterogeneous instead of a kind of homogeneous block here, you begin to see heterogeneity within the tumor. You begin to see new structures evolve and new contrasts come out. And that's because not all the cell sizes in the tumor are the same. And by going at these higher frequencies, we can distinguish between those to some degree because they have different ADCs. So normally when people are talking about measuring this diffusion property in clinical trials, they're making this one measurement, which is uniform everywhere, but in fact, depending on how you measure, you would get different results. So we thought, well, maybe because we're seeing new structure, and we think this new structure is associated with subcellular dimensional changes, maybe that's an earlier indicator of treatment response. So we show that here. This is before treatment, 24 hours after treatment, 72 hours after treatment. This is the conventional pulse gradient measurement of ADC with diffusion time of 30 milliseconds, standard clinical time usage. And you can see that we actually have some change over here, but not that much. If you do the diffusion measurement at 240 hertz, you immediately see a bigger difference compared to the background and substructures begin to evolve, such that we can say that there's heterogeneity here within the cell dimensions, and in fact we see earlier changes, earlier and bigger changes at these higher frequencies than at the lower frequency. So that was the first indication that maybe in a developing responding tumor, we were able to detect subcellular changes before there are gross changes in cell density and cell size. So that's what this says, that in principle we can find these small subcellular changes. We've done a number of other studies, but this was a very interesting one. We've done studies showing, for example, computer modeling predictions, etc., suggesting that, for example, if the nucleus changes size, it can be the main driver of a change of the ADC measured at these high frequencies, because the nucleus is sort of the biggest thing in there. So you can think of a model, for example, this is a particular drug, this aurora B kinase inhibitor, which when you treat the tumor causes what's called polyploidy. That is, you actually get doubling in the size of the nucleus, but it doesn't split. And you can get triple-sized nuclei, so it goes part of the way through mitosis but gets trapped. So it doesn't go all the way and split the entire cell. So you end up with cells with big nuclei, double nuclei, sort of, almost like Siamese twins sort of stuck together within a single nucleus. We did this treatment. This is the drug that actually inhibits this and causes this kind of effect. We made these measurements, and this is what you get. There's just two things to point out here. First of all, if you measure with the pulse gradient spin with the traditional method before and after treatment, you can see that we got the expected change, that in fact there was an increase in the ADC following treatment because we've begun to kill a bunch of cells, and we get an increase in the ADC. But if you go to high frequencies, we actually get a decrease. So what this is telling us is that at the high frequency, we've got more intracellular structure causing obstructions than we had before the treatment. So this is consistent with the idea that we are killing some cells. This is the other effect, which is that we're changing the intracellular structure enough to see a change and a drop in the ADC measured by this method. And in between, there kind of is not that much difference. So when you read a comment in the literature that the ADC dropped, that meant there was no response. You have to ask the question, at what diffusion time was that measurement made? It was made at a short measurement time. That's exactly what you would expect. So anyway, this is sort of an interesting area to now begin to look at how these ADC changes with diffusion time, cell density, and increased cellular structure. And we think it's potentially an earlier biomarker response to a certain kind of treatment. Okay, and now for something completely different. That was all about changing the contrast using external gradients, the coils wrapped around the sample that give you a gradient from the outside. Instead of doing it from the outside, you can also get intrinsic gradients, mainly from variations in magnetic susceptibility. So in fact, magnetic inhomogeneities occur at many different scales. For example, capillaries and veins have a different susceptibility to the surrounding tissue. And because of that, they disturb the magnetic field around them, and they set up intrinsic gradients of the field. And in fact, we've noticed in this Linus Pauling's work back in the 1930s that there's a difference in the susceptibility of blood, particularly of hemoglobin in the blood, according to how oxygenated it is. So we classify magnetic materials as being either paramagnetic or diamagnetic. Paramagnetic things line up with the field, diamagnetic ones don't. And oxyhemoglobin is diamagnetic, but deoxyhemoglobin is paramagnetic. So what does that mean? It means that if I do a little cross-section here, this is a cross-section through a microvessel or a vein. It's filled with blood. I apply a magnetic field. These lines of force should all be nice parallel lines, but in fact you see they bend at the periphery here because this material is different in its magnetic susceptibility to the outside. And that's the difference in susceptibility. And if you have a water molecule here and it's moving around, right, but it is not moving in a uniform magnetic field, it's moving from one strength to another and causing its signal phase and frequency to vary as it moves. So you have a bunch of these water molecules moving around, they will interfere with each other, and you get this transverse decay again. So you get this dephasing from the random motion of water molecules external to a magnetic susceptibility variation. And this depends on the field squared. As I said, it also depends on the blood. So if you have oxyhemoglobin, this is a line of force. It doesn't bend because it's pretty well matched to the surrounding tissue. The field uniformity here is going to be high, and the signal would not decrease. But if you have deoxyhemoglobin, you have these external gradients. That means the field here is non-uniform and the signal is attenuated. You say, well, how big can this be? I mean, it's a microvessel. Well, if you have something, let's say 5 microns in size, the intrinsic susceptibility of deoxyhemoglobin is about 0.08 parts per million difference. Sounds very small, but if you work it in 7 Tesla, that's a field of 5.6 milligauss. And 5.6 milligauss over a field of a few microns is 12 Gauss per centimeter, which if you go to an MR person, say how strong are your gradients, they'll typically say, wow, they're 4 Gauss per centimeter, or maybe 8 Gauss per centimeter. But 12 is actually pretty high. So these are significant effects, very significant effects, very similar to what we get when we apply diffusion gradients and look for signal changes. One of the interesting things is that this is now the basis for a technique that's been around for about 20 years called functional MRI, which I'm sure most of you have heard of. But it depends on the so-called blood oxygen level dependent signal, which says that signal losses, because of diffusion, depend on the oxygenation of the blood. If this is deoxyhemoglobin, then you get big signal losses. If it's oxyhemoglobin, you get small signal losses. When you have brain activation, like you might give some sort of stimulus, you get a change in neural activity. That leads to an increase in blood flow in the tissue locally. That washes out the deoxyhemoglobin. In fact, the brain becomes magnetically more uniform in that little region, and so your signal goes up. We call that the bulb effect, the blood oxygen level dependent effect. And it's widely used. This is an example of what happens if your signal changes, shown in yellow, from just tapping your fingers. If you tap your fingers, the primary cortex corresponding to finger movement lights up, supplementary cortex, etc. Very widely used in a number of applications in psychology and psychiatry. So just an example of how that's actually used, and I showed this morning, but we work a lot with primates. So this is a squirrel monkey brain. We take slices through the cortex, and these are 3 parallel slices. We stimulate the digits with these little piezoelectric vibrators, so it's a very subtle stimulation. When you do that, you can see the somatotopy in the brain, so these are the digits 1 to 5 in 3 areas of cortex with a resolution of about 0.5 millimeter, corresponding to those signal changes, those bold signal changes, from the fact that when you stimulate the brain, you wash out some of the deoxyhemoglobin, and the brain becomes more uniform, there's less magnetic non-uniformity, and you get a signal increase of about 1% or so. And this is widely used, and I've said that you can, for example, use this to study plasticity in the brain. So this is before an injury to the spinal cord, this is after an injury to the spinal cord. You can see that the yellow, which was not affected by the injury, is the input from D1, that hasn't changed, but the other parts of the cortex are remodeled because of plastic changes in the cortex following the injury. And this is being done in humans. Again, human studies at 7 Tesla, blowing little puffs of air onto the finger tips here, and then mapping those differences in the brain. So you can see here again the somatotopy of the different digits in this primary sensory area in the human brain. It's very useful for studying effects of injury, remodeling after injury, etc. So that's conventional mold. Just getting the movie. This is actually an Indian movie. These magnetic inhomogeneities occur at different scales. So they occur in capillaries as well as larger vessels. And there's been a problem in the fMRI field with distinguishing the effects from big vessels versus small vessels. Big vessels may be a little bit further away from the action, and in fact they dominate some of the signals, so they don't really tell you about what's happening in the cortex with microvessels. In addition, the dephasing effects due to diffusion contribute to what's both the intravascular effects, the effects that come from the signal losses within the blood itself, and the ones on the outside, which is to do with the water moving around outside. So people have thought of ways to edit the contributions of different size inhomogeneities. But this led to the question, the question we were interested in, what are actually the effects of diffusion on a signal if you have water molecules diffusing in a variation of the magnetic field? Well, this is where we're going to do a little bit more physics here. So we think we've got on the way to quantify some aspects of the intrinsic magnetic non-uniformities within tissues. Here's what we talked about before, that if you tip the magnetization, that's shown here, so it's after the V1 pulse, it lies in this transverse plane and it goes around at a certain frequency, and if the frequency is uniform, and if the spins don't move, it will just go around and around and around. But because they move, they actually see a little magnetic field that varies in time. So in one place they'll see a little positive field, then they move somewhere else and it's negative, and they will, as they move around from place to place, the field they actually see will vary. It'll go up and down, up and down, up and down. It's because of that time-varying field that you get this phase evolution and signal losses. And what it means is if you have a varying field, just like the field determines the frequency, this macroscopic magnetization, remember, comes from all these different contributions, different spins. They will each gradually fan out in this transverse plane, which they go at slightly different frequencies, different phases, and it's because of that that you get the signal loss. These all add up to a reduced net signal. So it's because of this problem. Well, suppose you did the following. Suppose you tip the magnetization, and then as soon as it's tipped, you apply a magnetic field in the same direction. That's to say, one that's in the transverse plane and also goes around at that same frequency. So we call that locking. That's called spin locking, where you tip the magnetization and you line up this rotating magnetic field with it. So when that happens, you've still got the fact that they move around, you've still got this bit. But the net field they see is no longer just this, it's this plus this, so it's actually that. And when they have this net field, which is varying a little bit, but it means they're processing not around this field like that, they're processing around this field like that. And that, just think about it in 3 space, what it means is you get much less dephasing. By locking the spins with a transverse field, you get much less dephasing, so your signal lasts for longer. We call this spin locking. The transverse plane magnetization, which is what we measured, still decays. Once you turn that field off, you can make a measurement. It has shrunk, but it's shrunk by less than it would have without the locking field. And the rate at which it decays, instead of calling that T2 now, we're going to call that something different. We're going to call that T1 rho, and its rate is R1 rho, which is 1 over T1 rho. So R1 rho is called the relaxation in the rotating frame, or spin locking relaxation rate. And it's less than you would get if you didn't put on this pulse. But the trick is, if you think about it, it will depend on the strength of this compared to that. If this is very strong, then this becomes negligible. If this is only small, then this still dominates. And so depending on how big this field is, you will get a different relaxation rate measured in that transverse plane. So that's what's shown here. What we predict is that spins will show an R1 rho, this measured time constant, this measured rate in the transverse plane, as a function of how strong that locking field is. And it will be high when the locking field is negligible, because then you're just back to the old dephasing T2, T2 star type thing. It'll be much lower when the field is very strong. And in between, there'll be what's called a dispersion, that depends on the strength of the field and some other properties of the medium. So we did a bit of theory, And here it is, the third of 4 equations, I promise you. Radiology, I didn't just begin to play now. Suppose that you have a sinusoidally varying field, that is one which has a gradient that goes up and down, spatially, and you can define a thing called spatial frequency as being sort of one of the wavelength, too high over the wavelength. So this is a distance, and so Q is the spatial frequency, one of the distance. And we have a gradient that's got some mean squared amplitude. What we predict is that the realization rate will depend on the gradient strength, this thing, the diffusion rate, and then how big this is compared to the frequency of the locking pulse, the amplitude of the locking pulse. So when the locking pulse is 0, then this would be 0, and we get basically a high value of R1 rho. As this becomes very, very large, this goes down, and that's what's described with this curve. Okay. The point is that this curve has sort of an inflection point, and that inflection point occurs when these quantities all form some relationship. In fact, that relationship is here, that at a certain value of it turns out to be a time, units of time. If we take the spatial frequency, compare it to the diffusion rate, we take that quantity, that's the relevant time at which we get an inflection point on this dispersion curve. So in other words, there's a point here that is characteristic of the medium in that it tells us this quantity. So if we measure a dispersion curve, if we take some sample and we imagine that within it there are these sinusoidal or time spatially varying fields, we can measure the realization rate on one row as a function of this locking field, and we'll find it does this kind of thing. It's actually a real measurement. We'll find this sort of inflection point here where the derivative changes sign, and from that we can work out this quantity, which tells us then this quantity. This is the scale of the inhomogeneity. So independent of how big those gradients are, we can make a measurement of an intrinsic parameter, which is the size of the variation of the magnetic field within the sample. What does that all mean? Well, for example, if you take little beads of different diameter. When the beads are, say, 1 micron, you pack them close together, your intrinsic scale for magnetic susceptibility variations in the sample is of the order of 1 micron. On the other hand, if you take 20 micron beads, pack those together, now your magnetic susceptibility variation is over a larger scale. So because of that, these dispersion curves vary according to the size of the little inhomogeneity, and in particular, the inflection points change. So in fact, this is that average so-called correlation time, which is a measure of this intrinsic property. And you can see it scales very nicely with the size of the sphere radius. So if we make a dispersion measurement, we can infer what the size of the intrinsic scale of those inhomogeneities are. And that's what's shown here. These are maps actually of this correlation time. These are maps of something about the intrinsic size of the objects. Without resolving the objects, we can say that this object contains something of the order of 20 microns, whereas this one contains objects only of the order of 1 micron, without resolving at that scale. So that's one of the aims we set out to do, is can we measure properties of intrinsic structure without resolving the structure. When you do this with brain imaging, for example, so this is somebody who's undergoing a breathing gas manipulation, so they become hyperoxic. So when they become hyperoxic, their oxygen tension goes up, you wash out the deoxyhemoglobin, and you get a signal increase, and what's shown in red are the signal increases that we would measure by different methods. This is the conventional method that would say that everything is changing, but a lot of this is actually dominated by large vessel effects. This is a conventional visual activation experiment where we're looking at V1, but in fact a lot of this is coming from large draining veins. All we can do is T1 row or R1 row measurement with different locking fields. And when you go to locking fields of say 400 hertz or even lower, you can begin to edit out some of these effects. In fact, this looks much more like true gray matter microvascular effects than do either of these down here. There's more evidence for this, which I'll just skate over, but another signature of how big the vessel size is that you're getting the signal from is how rapidly does the signal go up when you switch on the flashing lights if you're doing a visual stimulation. And you can see here that the grey echo goes up the fastest, so this is the big vessel stuff, and these go up gradually slower and slower. This is the R1 row sequence where we think we've edited out anything over a size of 10 or 20 microns. So this may be a way to improve imaging for fMRI, and in particular it may be a way that we can overcome some of the potential confounds in potential methods. If I have 5 more minutes, then I'll just finish on a topic that's really sort of not related. I should say, I was telling Dr. Figley earlier that some of these things are they may seem to be random topics, but there is one theme that ties them all together. These are all things I have not been able to get funded. So the things I have funded, I don't talk about. These are things that I think we should be funding. We talk about spin locking. But if you think about what's happening when we look at like this idea of a spatially varying field and things moving around between it, all we're talking about is spins moving from one instantaneous frequency to another. And there's another process that involves spins moving from one instantaneous frequency to another, and it's known as chemical exchange. That is, if you have a molecule such as a hydroxyl, you have a molecule that contains something like a hydroxyl, the hydroxyl is jumping on and off all the time. The resonant frequency of that proton that's in the hydroxyl is very slightly different, maybe 1 part per million, from the surrounding water. So it's moving all the time, on and off at some exchange rate between two species, two environments, which differ in their resonant frequencies. Amides do the same. Amides are about 3.5 parts per million from water, and the amide proton jumps on and off at a certain rate. And so those spins are constantly jumping from one frequency to another. That's actually been given, you can manipulate this in imaging. There's a technique known as chemical exchange saturation transfer, which I won't really talk about, but it's being used to try to isolate these labile species. And all we're talking about is that we have a spectrum here, 2 possible frequencies, F1 and F2. As they jump between them, then they will undergo a modulation of their resonant frequencies, and that will cause dephasing, just like we talked about. Well, just like we talked about, if you put on a locking pulse, so that the magnetization is locked in the direction of that transverse field, you will reduce the effect of this exchange, depending on whether the locking amplitude is large compared to the difference in frequency and how much they jump. These are simulations or calculations showing, for example, and also the other thing is, this difference here is not very big at low field, but it increases linearly with the field. So by the time you get to 7 Tesla, for example, 3 parts per million is 2 kilohertz difference. So that now becomes a very significant modulation compared to a low field. That's what's shown here. If I look at the effect of R1 rho, this dispersion curve against the locking field again, at low field, say 0.5 Tesla, 1 Tesla, there's not very much effect at all. By the time you get to 7 or 9.4, there's a significant dispersion curve. And it depends on the exchange rate, as you might expect. But the important point is here that, again, if we don't do a locking field, the relaxation rate will be high because of chemical exchange-based dephasing. If we put on a strong locking field, it's low, and there's a point in between where we can come up with a number that characterizes the exchange process. So, final 4th equation, I promise, is this one. This theory was worked out many years ago. This simply says you'll get a dispersion curve, and there will be a frequency somewhere around about here that's related to the chemical shift difference and the exchange rate difference. So if we measure this curve, and we find that inflection point, then we can come up with this quantity. And we know what this is, we know what chemical shifts are, so we can actually measure exchange rates. That was the theory. What we then realized is, well, we don't really want to measure exchange rates necessarily. We'd like to have images that potentially pick out species which have a particular range of exchange rates. So we call this the exchange rate contrast. I lied, this is the last equation. You have to write radiology. You tell them there's no equation, then you sneak them in on people. You take 4 images, one here, sorry, 3 images. You take one image here with this realization rate, with this value of the blocking attribute, one down here, and one in between. And you take the difference between this and this, this and this, and this and this, and you put it into an equation like this, and you get a curve. And what this curve says is this is the signal contrast in the resulting image according to how the exchange rate relates to this number. So if you're at this exchange rate, then you get a big signal. But if your exchange rate is much different, you don't contribute much at all. So you can think of all these protons in a tissue in multiple different environments, and think of there'll be one group whose exchange rate is, say, 500 Hertz. So by choosing the right frequency here and doing this manipulation, you can get all the spins which have an exchange rate of 500, and maybe those including, say, 300 and 600, but things that exchange very rapidly or very slowly won't contribute. That was the theory. This is one example of that. We actually looked at these exchanging protons, and this is actually X-ray contrast agent that's FDA approved. And this is showing this exchange rate contrast when we pick out that different intermediate frequency. We can isolate this example, which is just the iohexol. We can move it up higher, and we can accentuate this, which is now the iohexol with BSA. And then this is moving even further, where this is the iohexol. This is now just BSA on its own. So this told us that we could, in fact, manipulate contrast according to the exchange rate of the protons. And then we did it even further. This is 3 different polypeptides. Ignore this. This is the result using CES. But 3 different polypeptides where we've now got this exchange rate contrast. We've picked out 3 selected values for that intermediate frequency. And we can isolate these according to where we chose that intermediate frequency. So even though they have very similar chemical structures, some of these structures, their detailed exchange kinetics are different. And if you imagine an environment, for example, where the pH changes, those would be even more different. And so this could be a way to actually accentuate species which have a particular range of exchanges, which might need something in terms of the underlying biochemistry. Finally, using that same equation, if I look even at 3 tests, if I look at R1 rho as a function of the locking field in cartilage, I get a nice little dispersion here. I can fit that to these parameters, and I can actually make a map of the exchange rate of the protons in cartilage. And we know that most of the exchanging protons in cartilage are the hydroxyls, the proteoglycans, and those are changed in things like osteoarthritis. And people have been chasing how to image cartilage forever using different techniques. Maybe doing this quantitative measurement of exchange rate would be one way to address that somewhat differently. So just to summarize, in NMR, a great deal of effort, I didn't talk about this at the beginning, engineers and companies spend a great deal of effort trying to make a magnetic field as uniform as possible. The MR machine that you buy has a magnetic uniformity of better than 1 part in 10 billion across a very large volume because of the construction and all the rest of it. And then, we like to screw it up. We like to use the effect of field inhomogeneities and gradients to make that field non-uniform in a different way. And one way is to look at tissue microstructure using things like diffusion or intrinsic susceptibility, things like fold effects. And we're trying all the time to get information below the spatial resolution of the voxel, and MRI really lies in the ability to understand these kinds of contrast mechanisms and use them for new purposes. Finally, these are the people who actually did the work. These are all been graduate students of mine. These have all been postdocs. And I show you our building. So here's our building. It's always Christmas in Nashville. And as I like to point out, good students of postdocs are always welcome. The other kind, stay here, but by all means, if you're ever interested in like moving, send me a resume, drop me an email, or all of that. Thank you.
All right, I think we have time for a couple of questions. Oh, yeah, watch your eyes, sorry. couple of quick questions if anybody